Hi, everyone. Hope you are all doing well. Thank you for joining today's webinar. Today we have an awesome panel, and individuals are going to talk a little bit about their role, their experience in the field of clinical laboratories. And so we will introduce the panel members just a bit later and hear from all of them. I'm just going to give everyone a few minutes to join. As a reminder, these webinars are recorded. We do put them onto the ClinGen YouTube channel, and we have a playlist for that. We will put that playlist in the chat. This week's recording will be up by the end of the week. The recordings from last two are there. So the first webinar we had on the 18th, I believe, was LGBTQA and genetics professionals. These individuals discussed their experience as members of the field of genetics. Last week, we had a panel of genetic counselors go through their experience. And then today we have clinical laboratory career opportunities, and the rest of the schedule is on the slideshow, and the QR code is to the playlist for all of these recordings. As a reminder, everyone is able to ask questions. There should be a Q&A box that is at the bottom of the Zoom. It's more to the bottom right of the screen. You could type in your question. If someone else has the same question, you can upvote. So this just means likes the question, and then we will try and get to them during the panel. Before we begin, I just have two quick questions that I'm just going to ask everyone, and I will start the poll in 1 second once I find the correct button. So the first question just asks, as of September 2024, will you be a student, and where will you be a student? In which area? Will you be in high school? Will you be in your undergraduate? Will you be in your master's program? And then the second question is, how familiar are you with clinical laboratory careers? So give everyone just a few seconds to answer those questions. Oh, you guys are so fast, so thank you so much. We have, looks like the majority of individuals are currently not a student. We have about 20% are PhD students, about 16% are undergraduate students, and a few are in the master's program. And we have some people who are not familiar with this field at all to individuals that are very familiar. So just taking a look there. Fantastic. Okay, so just a few slides of information. What is a clinical lab? It's a lab that performs testing on humans and returns results that are added to the patient's medical record. Labs are required to maintain different certifications, and this is encouraged to promote consistency, quality, and transparency in the laboratory testing. Currently, there are over 260,000 CLIA certified laboratories. Examples of clinical laboratories, many work together, overlap, but there could be a microbiology lab, a molecular genetics lab, cytogenetics lab, pathology lab. Sometimes one lab does it all. And right now, in the US, testing is regulated by the Centers for Medicare and Medicaid Services, but there might be some changes coming in the future. So what's the difference between a clinical lab and a research lab? So clinical lab is indication-based, so returns results to doctors and patients. There's standards required for maintaining those laboratory accreditations. Specific certifications and training are required for the certain labs. There's structured validation procedures for new methods, machines, programs, procedures, up-to-date procedures and SOPs that are all stamped in terms of when that SOP on the date that it went into effect. And there's lots of sample documentation that is needed. Research labs are more discovery focused, disease research or basic science. It could fall into many different lines of work. So humans, cell culture, model organisms. they require their own ethics board and approval, so sometimes an IRB approval or an IACUC. Experiments and protocols can be changed and modified, so it's pretty fluid, right? We're learning the new procedures, they're doing the research, often funded by external grants, and so such as grants from the NIH or various foundations. And results return is contingent upon study design. So not all research studies will return results. Some research studies determine a date that they will return results, and it's all based on how the study is set up. So these labs are not mutually exclusive, and research studies often do take place in CLIA certified laboratories. A wet lab are personnel who are physically in the lab, so receiving and accessioning the sample, performing the test, running the quality controls, and then a dry lab is a person who participates in the process outside the laboratory, so the results in interpretation, bioinformatics, software design and development, quality control, returning the results, drafting the reports, etc. And there is a wide range of career possibilities. So some individuals will have a specialty. So maybe they'll work in molecular biology that is geared towards prenatal disorders or diagnosis, or maybe metabolic, or maybe some individuals will work in microbiology, focusing on other areas. And there is also a wide area of specialties and variable set of like employment settings. So some people will work for a university or a hospital, will work for a non-for-profit or a profit laboratory, and then other government organizations. And here are a few additional resources. The American College of Medical Genetics and Genomics is a big source, Association for Molecular Biology, and there's different training options for geneticists. So you can go down a route of becoming MD, a PhD, or the MD PhD route. So we have a lovely panel for everyone today. I am going to stop sharing my screen and we will start introducing the panelists. I ask that the panelists will start by just saying who you are, where you work, what you do, and then if any other panelists have a similar path, you can say that it was similar or chime in with some of the little bit of differences. As we mentioned before, I'm just going to go in the order that everyone is currently on my screen. Rachel, I'm going to start with you.
Sure, thanks. So my name is Rachel Burnside. I work at the University of Florida. I'm the lab director for the cytogenetics lab of the Department of Pathology here.
Fantastic, thank you. Alexa?
Hi, I'm Alexa Dixon. I work at Washington University in St. Louis School of Medicine. We have a diagnostic laboratory here that does molecular diagnostics. I spend much of my time analyzing patient samples sent for exome and genome samples testing.
Thank you. Amelia?
Yes, hi, my name's Amelia. I work at the National Eye Institute, and we have a clinical lab there that specializes in ophthalmic genomics. So I'm a biologist there. So I started out as a technician, and now I'm doing more, developing assays and some translational research as well.
Thank you. Scott? Scott, you're still on mute, so I'm sorry.
Sorry about that. I'm Scott Smith. I work at SUNY Upstate Medical Center. It's a hospital setting. I'm a director in the cytogenetics and molecular genetics laboratories.
Thank you. And Benjamin?
Hi, I'm Ben. I'm assistant professor at Vanderbilt University Medical Center. So same as Dr. Burnside and Dr. Smith, I'm also lab director at here Clinical Molecular Diagnostic Lab and Cytogenetics Lab. Nice to meet you guys.
Thank you all. So now I'm going to ask a few questions about how you found your career. So how did you learn about your career? How did you decide maybe what school to go to? Or I know that a lot of people sometimes have like a winding path until you got to where you're at. So maybe you could share a little bit about what pulled you into the directions that you are currently working in.
We're going in the same order?
Yes, sounds
All
great.
right. Yeah, so I definitely had a winding path. And I was a technologist after I graduated University of Houston a million years ago. Basically, I had no idea what cytogenetics was. It was, I got a job. I was super happy to have a job. And I learned what cytogenetics was on the job. Knew I wanted to go to graduate school at some point, but I needed a break because I'd been in school for a very long time. So I worked in the lab as a technologist for 2 years, and then went to the University of Kentucky for my PhD, and did not know how to become a director at that point in time, because when I was a technologist, my laboratory directors had PhD clinical genetics certification, which was no longer offered. So I had considered doing a research setting using cytogenetics, but I didn't really want to do research. And so those were, the early days of Google. So I was Googling different options and ran across the ABMG postdoc for clinical cytogenetics. And that was that. The rest is history.
Yeah, I had a very different experience. So after undergraduate, I went straight to graduate school with entirely the intention to become a professor. I loved research. I thought it was so fun. I did a postdoc after graduate school and then couldn't find a job. I just couldn't find a professorship. And so I just kind of fell into the position that I have now. They were hiring for someone to help, determine the clinical significance of all of these DNA variants that they were detecting in a clinical lab. And so I applied and I got the job. And really what's been awesome about what I do is that in every position that I've had like this, I've been really able to tailor it to make the job kind of what I want. As Amelia said, at one point I was designing tests to, offer to the public, and that was really fun. I've been involved in clinical research, translational research, and I've done some teaching. So it's really been fun to be able to make this job what I want it to be.
Thanks for sharing, you two. I am a little bit of a mix of the beginning of both your career paths, it seems. So this job, I graduated with a degree in biology from the University of Maryland. I knew I wanted to work in a lab. I knew I wanted to do something related to research maybe, but I wasn't really sure. I loved the lab setting. So I had trouble finding a job. With just the undergraduate degree, and a recruiter actually ended up reaching out to me. And so I began as a contractor working in the same lab that I'm in now. So starting out like we were talking about at the technician level. So, accessioning, processing samples, doing some, Sanger sequencing, very basic level genetic analyses. And after a couple of years there, I felt like, I was getting in, I was ready to take the next steps. So I decided to get my certification in molecular biology. And that really gave me, the extra knowledge and support to kind of expand what I was doing and grow our lab and, develop new instruments to implement as well. And then I'm currently pursuing my master's degree in biotechnology at the moment. So I'm halfway done with that. So I'm still, working my way up the ladder.
So I was, when I discovered this field, I was doing my second research postdoc, and I had been informed that I was out of funding. My PI couldn't help me. I didn't have a grant coming in. I was looking for opportunities. I had applied for a couple of faculty positions, mostly at teaching institutions. I didn't get them. And I had a friend who had just gotten into a clinical chemistry fellowship, and she was telling me about her path. And I thought, well, maybe there's something like that for genetics. And so I did a little hunting, and I thought, well, gosh, this is exactly what this is perfect. I'm going to do this. And I set about applying to the ABMGG fellowship program right then, and fortunately was lucky enough to get in on my first pass, which isn't necessarily the normal route anymore, unfortunately. And 3 years later, I I had a job as a associate director, and have since moved from that position, but very happy in this field. I think it's an amazing field. I really enjoy it.
Mine was slightly different, so maybe I can start with my PhD experience. So, for my PhD, I studied for Marfan syndrome, so much more clinical human genetics I studied. So my research was purely based on basic science, but my PI was MD and he runs his own Marfan syndrome clinic at Johns Hopkins Hospital. So I was able to participate in his clinic, and I analyzed tons of patient data. And then one of the key data was patient genetic testing results. And then every single time I read it, there's somebody who signed out the case, and they have something special letter at the end of their name, which is FACMG. And then later I was curious about what that it is. So I searched it and then realized that there's kind of title for the lab directors or fellow of American College of Medical Genetics. And I realized that that is qualified for the PhD as well, not only for the MD or MD PhD. So I searched about it and then I talked to many other FACMG people who working as a lab director. So after that, I decided to pursue this career path. And then after that, same as others, like doing the fellowship and then took the board exam. And then now I'm settled out here. So I think the key thing is maybe you can experience the clinical stuff that would help you to choose your career path in the in the future.
Great, thank you. So I hear that maybe the clinical experience was really great to have before you applied to your graduate program or to your fellowship. And so I was wondering if you, I know Dr. Smith mentioned that now some folks don't make it into the fellowship on the first application. So maybe if you could expand a little bit of what you think would make a good candidate or a good applicant. What are some of the maybe prerequisites or some of the things to do to gain exposure in order to make yourself a competitive candidate as you apply to the grad programs and apply to the fellowships?
Yeah, that's that's a really good question because it is very competitive to get into these programs. When I came into the field, cytogenetics and molecular genetics were still separate fellowships. So now they're combined into a single fellowship. And anybody who knows their chromosomes automatically has a leg up. So the fact that I was a technologist in a cytogenetics laboratory and knew my chromosomes really gave me an advantage, even in a separate cytogenetics and molecular genetics setting. But even now that they're combined, it's even more competitive, right? So, you know, just again, having been a technologist and having that knowledge already was very helpful to get me in. I think I would just add to that, just being curious about genetics is a good trait to have.
Yeah, I would agree. I guess I'm speaking out of turn here, but I would agree. I think I feel I was very fortunate, really having no knowledge of the field at all. I took a real chance, and they took a chance on me. I think having some familiarity with the field isn't going to hurt you by any stretch. I guess I did have a number of years as a postdoc in research, primarily in cancer, and most cytogenetics laboratories now are very heavily cancer focused, and so you know that may have helped me a little bit, but I think the more you can shadow in labs, the better off you're gonna make yourself if you're looking for a career and I agree with Rachel 100%. Like curiosity in genetics, you know, it's not gonna you're looking to spend your career in genetics. You better have some curiosity about it. So I agree with that 100%.
I think what's awesome about this field, though, is that you don't have to do a fellowship necessarily to be involved. So it sounds very much like Amelia and I both have taken a similar path in that I don't particularly want to do the fellowship. I've had a professional job now for 10 years. I'm not interested in more kind of fellowship training. So there's lots of opportunities to get into the field either as a technician or a technologist, or as I did as an analyst, and kind of move up or move around. Similar to Amelia's experience, I got an ASCP certification in molecular biology, just to kind of show that I was interested, that I had curiosity in genetics, and that I was dedicated to the field. So if you're not interested in spending more years in training or fellowship, there's lots of opportunities to get involved.
Maybe I can go next. So I already mentioned that the clinical experience is so important because program directors always looking for the candidate to understand this about the fellowship or about this job. So if you do not have any experience, it's hard to get in the fellowship or even hard to get the permanent job. So I recommend you, if you're postdoc or students, take an experience in the clinical lab and then just understand what's going on over there, and then whether it is kind of fit with your career path or not. And then number two, I realized that is so much teamwork based, like compared to research one, I think it's much more teamwork. Like as just Alexa mentioned, like always multiple people working on their position. Without their help or without this teamwork orientation, it's hard to achieve our patient care service. So if you are thinking about yourself and if you are a team worker or team kind of spirit, that is kind of right path for you. I highly recommend that. And then think about that. If you are not so much teamwork person, then think about something else, I think. Yeah.
That is great information to have and great information to share, because I'm a genetic counselor and I rely on everyone on this panel in order for me to be able to discuss any results that I get with my patient. I need to go through all of you. I need to ask all of the questions. I need to put all this together. So definitely teamwork. Everyone needs to put their heads together and put all their expertise together in order to provide the best patient care. So this question may be a little bit hard. So I was going to ask why you think that your roles are specifically different than other careers in genetics. So if you were going to sell someone on why your career is the best in genetics, what features would you think you would come across or really like to highlight?
I can go first on this one, especially since, working at the NIH, there's an aspect of research that we do, but I think what really separates the clinical genetics laboratory versus other genetic research laboratories is getting to help people, right? Like that's totally, it really comes down to feeling like you're making a purpose, or you have a purpose and you are able to help provide answers to these families who, otherwise aren't able to get answers. So at least that's for me, that's definitely one of the biggest differences, but it's also the biggest reward as well.
Yeah, I think that's a great answer too. I would say one of the things that is probably common personality trait for a lot of people who are working in the laboratory is that we like to solve puzzles. And the puzzle just happens to not be, it's related to an individual, but we don't have direct interaction with patients. We're, dealing with their samples, their blood, their tissue, whatever. But yeah, we are very much puzzle solvers.
I'd agree with that. And I'd build on it to say the other thing that I think maybe sets us apart is I enjoy continuous learning. Every day I learn something new. And I think it's a really great opportunity to apply the knowledge that I already have and continue to build on that. It's really fun.
For me, it looks like there's two different things between the research lab and clinical lab. So research lab, they focus on one question for many years, 5, 6, 7 years. But here, clinical lab, every single day, you have tons of new problems, new cases, you working on something new, and you identify something from there. So it's totally different environment. And then again, it's kind of teamwork based one. And then another thing, if you're willing to help somebody, like actually help to provide patient care service, because based on your test result, the patient care service start from there. So if you are willing to help people and then we are kind of more, what can I say, clinical mind, then I think it's much more appropriate for this career path.
And I agree with everyone there. I think I would just add that like I have sort of a short attention span sometimes, and so having the variety come through is ideal for the way my brain functions. And so being able to contribute to science in a research capacity, but then also from a clinical side, I think is one of the perks for me as well. So.
Thank you. And I know multiple of you have said that it changes every day what you're doing. So if you could give broad strokes, like what would a day in your life look like if you were to go in and start your day? What are some of the things that you could encounter? And you know, maybe not do a day, maybe average it to a week of what you would experience.
So I've worked in a large commercial reference laboratory for a long time, and now I work in an academic laboratory, and they are completely different. My day in a large reference laboratory was primarily case sign out. It's a lot of volume, and if you have academic interests to write papers, you know, you have an interesting case or a series of interesting cases, you kind of just have to fit that in during the course of your day. Your teaching takes a different direction. It's more oriented towards educating your technologists and your technical staff than it is actual students. In an academic laboratory, my day is very different. So, and it's a smaller laboratory too. So I have a lot fewer cases that I deal with on a day-to-day basis. Now I have responsibilities for teaching primarily medical students, but I have opportunities to teach undergraduate and graduate students as well. There's obviously the expectation for publication in an academic institution that you don't have in a large reference laboratory. So it's a little bit of a mixed bag, but I also feel like in this setting of the academic laboratory, and part of it is where I am in my career too, I have a lot of freedom to determine what it is I want to work on outside of casework.
Yeah, my experience actually has been very similar to you, Rachel. I started at a larger reference laboratory, and it was case volume a lot, but also some development of new tests that we were going to offer. And now working at a medical center, a medical school. It's still every day I do cases. I've done 2 this morning so far, but I'm also working on a couple of different publications, like Rachel said. I'm also teaching. We do graduate students and then also fellows for the ABMGG fellowship program. And so, yeah, it's been fun to do a bunch of different things, have my hands in a bunch of different kind of aspects.
Yeah, for me, it is, you know, a lot of maintaining the lab and the equipment is one of the biggest things that, and the documentation that goes along with that. So a lot of documentation is probably one of the big things. Updating SOPs that need to be updated, which is crucial. You know, every little tweak needs to be reflected in our protocol. I do a lot of training as well. So, you know, getting technicians up to speed so that they can do more complex testing. Our lab also participates in the Laboratory of Genetics and Genomics Fellowship. So we do a lot of training with, you know, people coming through, spending a few weeks with us, and that's always really rewarding. And then experimental design. So, you know, making sure we have the samples, they're diluted the right way, you know, our primers are ready, you know, just all of these things that need to be done before you can actually do the experiment or do the test. So a lot of those types of things.
Yeah, pretty much the same thing. So I'm also working at the academic medical centers. So I have 3 doors. One is clinic, and then the other one is research one, and then the last one is education. So yeah, pretty equally, I spend my time for 3 components. So clinical case, so clinical case, I signed a clinical case daily base, like in the morning and then afternoon, and also sometimes in the evening for stat case. And then for teaching, I usually teach resident and then fellows, clinical fellows at here in Department of Pathology, and also teach some grad students and also genetic counselors. Counseling students, not counselors. That's what I teach them. And then also lastly, the research is slightly different. So my role is much more focused on the R&D stuff. So developing new tests and then validate the new tests. That's kind of my primary role. And also I collaborate with some basic science researchers. So also participate in them and then work with them for the basic science research.
Mostly already been talked about. Whatever, same thing for me. Sign out cases, molecular and cytogenetics. Again, I said cytogenetics mostly is cancer, at least at our institution. I've got some leadership roles in the laboratory, obviously, that people are looking to you to make decisions, and then teaching primarily residents here, but also fellows and undergraduates and medical students. And then each of us is expected, again, at an academic institution, each of us is expected to have some sort of research protocol in place and publication metrics. Nothing new from what's been talked about already.
No, that's all great information. So thank you for sharing. We have a few questions that we received. So the first question is, what about remote job opportunities from individuals in another country in this field? And so maybe not remote job opportunities, but maybe what are some opportunities to learn more about the field? And maybe talk a little bit more where you would gain experience. I know you guys all had a different winding route to find it, but is there any information that you all are teaching students? So what do you guys share with them in order to gain exposure or recommend in terms of shadowing? Are there any resources that you have to share?
Yeah, so I actually am working on getting an undergraduate here in my laboratory who is interested in learning more about clinical laboratory science. This is a heavily regulated field, and I work in a state that requires licensure for the technical staff. So without that, you're kind of limited in what you can do with respect to handling patient samples. But that's not to say that there's not a key role that these students can play in the laboratory with respect to making media or helping accession samples or just doing what needs to be done to help the lab run smoothly without actually doing the experiments themselves. We also have a junior college here in town in Gainesville that has a medical technologist program. So we will get those students, cytogenetics is a totally separate field that is not part of the medical technologist degree per se. So a lot of them don't even know what they're getting into when they come to our laboratory. They spend half a day with us and they've got the deer in the headlights look of like, what is this that I'm doing? And then by the time they leave, they're very excited about genetics. So I think we're doing something right here with the med tech students. So yeah, I mean, if it's an interest, especially in undergrad or even in high school, there's age requirements for being in a clinical laboratory too. But if you meet all of those, just express an interest, and where there's a will, there's a way to get in and get exposure.
I think the other way that you could gain more experience, if you have a bachelor's degree or an undergraduate degree, the Clinical Genome Resource, or ClinGen, is always looking for biocurators. So joining expert panels, learning the SOPs that ClinGen has, which would give you great experience in just following an SOP and how that kind of looks. And then also learning more about these specific expert panels and what they focus on. And anytime we're hiring here at our laboratory, having ClinGen experience on your resume is a real bonus.
Yeah, Alexa stole what I was going to say. So yeah, the ClinGen expert panel is definitely a great way to get involved. They're always looking for volunteers. I will say the ASCP, the American Society for Clinical Pathologists, is a great way to kind of get your foot in the door as well into a clinical lab. They have some requirements to be able to sit for the test, but, it could be taking classes. I took the non-traditional route because I was already working in a clinical lab, but that's something that you can look into as well.
Yeah, there's some kind of trend new in these days. So for technologists or clinical scientists, there is some kind of much more remote options. So if you are looking for the remote option, there's a tons of remote option jobs, so you can apply for that. And then that's much more like corporate jobs or commercial clinical labs, they provide that option. But in these days, especially my institution, Vanderbilt, we also provide some kind of hybrid one. So initially, we need some in-person training, like a couple of months, and then later you can switch to the work from home or work remotely, or occasionally you come to the lab and then do some kind of in-person work. But we try to provide that, and then that's kind of trend. So everybody's looking for the work from home or some kind of flexibility. If you have any family issue, or even if not, you want to work some more flexibility. So I can see that's the trend. So if you're looking for that kind of job, then I think you can, easy to find it.
I also live in a highly regulated state, so this is New York. We have beasts of laws that dictate to us what we're going to be able to do and not do, including we have to submit our validation studies to the Department of Health and get their approval before we can implement them. So that's unlike any other state that I know of. Maybe Florida's the same, I don't know, but it was a shock to the system when I came here. I knew it was going to be an issue, but then being confronted with it gives you pause, I guess. So we have requirements for the technologists. There are requirements for the methodology we use, but there's in most labs, at least, including ours, you can often get on-the-job training. So the state will give, if you've got a bachelor's degree, you have passed their evaluation, which shows that you've got coursework in the basic sciences. Technologists can train with us for a year and then become licensed. That's not the same for someone who's for example, a variant analyst. Those may or may not require a PhD depending on the organization. Having some sort of certification isn't going to hurt for sure, but it's not necessarily required depending on the organization. Certainly not from a state perspective that I know. So there, as Rachel said, like there's a will, there's a way. You can find your way in if it's something that you're interested in.
Thank you. And I think going along with the will that there's a way, I think a lot of us here really rely on networking opportunities. So talking to people, different meetings, you never know where that opportunity will present itself. And so as a ClinGen employee and someone who will plug ClinGen even more, volunteering to curate with a number of these biocurators, number of these researchers, number of these physicians, number of these lab directors, it really helps to gain exposure. It helps make connections. It helps broaden your network and to hear, hey, we're looking for someone to help out with this. We're looking for someone to help out with that, and really able to expand your opportunities. And ClinGen does support people from all over. I think we have members from 65 countries right now. So continuing to expand, which is fantastic. There are a number of questions we received regarding the fellowship. So I'm not sure if some of the individuals who did the fellowship pathway can talk a little bit more about what it looks like in terms of the education, the rotations, the classes. I know we talked a little bit already about what to make a standout or a competitive candidate.
Yeah, as I mentioned, the fellowship has changed since I went through fellowship, although the basic mechanism of what it is has not really changed. It's just now combined cytogenetics and molecular genetics together. There's a didactic component. Some programs are 2 years, some programs are 3 years, some programs require a research project, some do not. There is a requirement, and the board actually will lay out for your logbook. You have to go through the clinic with the physicians and see so many patients. You have to do so many different kinds of assays on the bench. So you're a little bit of a glorified tech in the laboratory, although you're not really, because you're a postdoc. So you really are immersed in both the technical side and the clinical side during a fellowship.
Go ahead, Scott.
Thanks. I was just going to add that, you know, the didactic component, you know, not only are you immersed in the clinical side and the technological side, but in the theoretical side, I think too, because you are in your spare time trying to memorize textbook after textbook after textbook for the board exam, which is the big pillar at the end of fellowship that we all put a lot of weight on, obviously. And so really, for me, fellowship was a lot of fun. I learned a ton. And again, I didn't go into the field having previously had experience in clinical cytogenetics or clinical molecular genetics. And so everything was just, I was a sponge. It was great. And then the exam comes and there's pause there. It's a trying situation. It can be a trying situation. But you know, people can get through it. And most people do. And so just know that that's coming. If you're going to fellowship, know that that's coming and plan ahead for it, because it 2 or 3 years sounds like a long time, but it really isn't. Especially if you're trying to learn a lot of stuff, which you would be. So
Yeah, so I think I'm the most recent graduate from the fellowship, so I can speak a little bit more about it. So it changed a lot. So last couple of years ago, ABMGG kind of changed their program. Now it's ACGME certified program. So maybe you can look around what that means, but even still, I don't understand what that is. But anyway, it's changed a lot and then much more centralized, and still some depends on each program is slightly different. Some program more heavily focused on technical skills, and then some program much more heavily focused on the medical knowledge or clinical genetic knowledge, or some lab much more focused on lab management type of thing. So yeah, you should contact each program director and then ask about their program, and then you can find your ideal program. And then basically the basic structure is they provide, well, first year, they provide 2 didactic courses. So maybe you can sit down with graduate student or sometimes genetic counseling students, and then took 1 or 2 courses. It's much more clinical genetics related courses usually, but sometimes they also provide basic human genetics courses. And also technical stuffs, like first month of each rotation, you may need to spend tons of time on the bench, and then they will treat you or train you almost same as the other technologies. So you should be able to independently perform the assay. And then later it's much more focused on analysis and then case sign out how to draft the report. And then another component of the training is much more like lab management and the QC, quality control type of thing. So what kind of inspection you need to do, and then what kind of QC metrics you need to follow up, and then how do you manage your lab, and then how to organize, operate the lab type of thing.
Thank you. And Alex, I'm going to gear the next question more towards you. And it says, can you weigh in if there is any differences between variant curation and variant scientist position? And are there positions that would be available straight from a PhD? And so this seems a little bit more similar to your career path.
Yeah, definitely. Yeah, like I mentioned, I was not interested particularly in doing additional training after I finished my PhD and my postdoc. So yeah, there are lots of opportunities as biocurators or as variant scientists or variant analysts to be able to like get your foot in the door kind of thing. So yeah, it's definitely an opportunity. I actually did an analysis of kind of the workforce in general, and there's a lot of different jobs. They're called analysts, they're called scientists, they're called biocurators, there's a lot of different titles. So really, you just have to kind of look around and careers.com or those sorts of things, or LinkedIn, like you mentioned. LinkedIn is a great way to kind of look around and find things. And like Caitlin mentioned, networking. I actually found my job at a bar. I was telling someone that I needed a job, and he said, "Oh, I know somebody that knows somebody that has a job that sounds like could be for you." So he contacted that person. That's how I got my job. So networking does play a big role as well. So yeah, there's lots of opportunities if you're not interested in doing additional training programs, official training programs. Um, yeah, just look around.
Thank you. And an important question that someone asked, and I think it's all good to know before you pursue different careers, is can you comment a little bit on the work-life balance and how you all are able to manage work life, personal life, home life, career life?
Yeah, we're all, I can probably speak for all of us and say we're Uber nerds and can really get into our work. But I always, especially when I was at the commercial lab and it's a 24/7 operation. I had conversations with colleagues who wanted to have a clean desk before they left. I was like, it's not going to happen. The work never stops coming. You just have to cut it off and go home and spend time with your family, right? And nobody at the end of their life is going to say, oh, dang, I really wish I worked more, right? That's not where we are. So you really have to be disciplined about calling it the end of the day and going home and spending time with your family. Plus, having hobbies that you enjoy doing, you want to create time for those as well.
I will say one benefit of kind of the clinical laboratory versus the research laboratory is that, I didn't have to go in and check on mice on the weekends. I don't have to go in and feed cell culture on the weekend. So while Rachel mentioned you could make it a 24/7 job, I think it is important to have those kind of cutoffs. And there are some that are, a little bit easier to make as a clinical geneticist versus a research scientist.
Fewer time points, that's for sure.
Yes, exactly.
Our laboratory does have a Saturday rotation, but the technologist who works on Saturday gets another day off during the week, right? So Saturday is really a light day. They just come in, they take care of the cell cultures if they need to, or do a harvest of cultures if they need to. But then, they're not expected to work 6 days that following week. They'll have an extra day off. So, and the techs rotate that. So maybe once a month, once every 6 weeks or so, you'll work a Saturday. I don't work on Saturday.
Yeah, just second what everyone said. I don't really have anything extra to add, but you definitely need to make your own boundaries. You could work forever if you wanted to, but again, in the end, what does that do for you, right? So gotta know when to close the computer.
Yes, I started working with mice and I quickly said I would like to become a genetic counselor. They were not as fun. My last question will be for some of you to kind of think about the future. Where do you see your career in, 5 to 10 years? Do you think of any changes or education that you are trying to get on ahead of as we move towards, the future in clinical genomics?
Yeah, so I'm actually the last cohort to be doing the alternate path for laboratory genetics and genomics. So I'm working on my molecular logbook. I actually just turned it in last week to the board. So I sit for one more board exam in my life, and hopefully I never have to sit for another board exam. But yeah, I mean, my goal is to have a really fully integrated genomics laboratory. So not just cytogenetics, but also molecular genetics. We do have molecular pathology here that focuses primarily on oncology. So the opportunity for molecular genetics is constitutional testing here in my laboratory. So really looking to grow the business and to grow my own skill set.
I think for the field in general, one way that I'm seeing the field in general moving is towards AI, and I think that the early adopters of AI would probably be the ones that will be most well positioned to utilize it and to kind of grow it the way that we want to grow it. So that's one thing that I'm very interested in, is learning more about AI and how to integrate it into our processes and make it work for us. And one aspect of that is that I think having coding experience would be really useful for someone like me. So that's something I'm looking into, and would encourage anyone that's interested in this kind of field to get some experience with coding and knowledge about AI.
Yeah, I think in terms of the field, it's definitely going to be an integration of all of the big data. You know, we have all these EHR, all this data from EHRs, you know, the DNA studies, and then incorporating RNA studies and epigenetics and cytogenetic studies all into one to really solve these way more complex cases that basic DNA sequencing isn't able to solve alone. So that's where I anticipate it going, and I'm excited for it.
Yeah, one thing I want to mention is there's now in this stage lots of targeted therapy available, and that it will become more and more available. And then when you want to treat your patient with targeted therapy, the genetic testing is always necessary and mandatory to treat the patient. So think about that way. I think that this field will grow and more, and much more routine tests for the cancer patient or any other genetic disease patient, I believe. So yeah, keep in mind that.
I'd like to think that the field will stop hollering that cytogenetics is dead, because it's never going to happen. And so I wish those who use their voice to say that would cease from doing that, because it just has been detrimental to especially the cohort of technologists coming into cytogenetics. Because there aren't any. It's people who have been were trained many years ago, and the groups that are coming in are thinking that this is not a field that they will be able to survive. But instead, I think what's going to happen is that, as everyone has mentioned, there's going to be much more data to be looking at that needs to be integrated, rather than we're going to get rid of this group of data and get rid of that group of data. Cytogenetics is important for what it does. You don't need to order a next generation sequencing to rule out Turner syndrome. You don't need to or Down syndrome, or any sorts of things like that. Like that's just silly. And so I'm hoping that there will be a new realization that each one of these specialties that we all use to help diagnose patients will become that much more evident of importance and be used for what they are being necessary for. So that's where I hope to see the field. I don't know. We'll see.
I tell our medical students, it's like an MRI does not solve all your imaging questions. DNA sequencing does not solve all your clinical questions.
That's absolutely right.
You're
That's
absolutely right. Cytogenetics is not dead, and it's
Yep.
not going anywhere.
And everyone should learn your chromosomes.
100%. Yes.
Yeah, and you can go to what is it, the Wisconsin website. Oh gosh, now I'm going to forget the name of it. Anyway, there's a karyotyper. Look up karyotyper, and you can learn your chromosomes in a digital game format. It's quite nice.
Fantastic. Well, thank you to all of our panelists. We all really appreciate your time, appreciate sharing your experience and your expertise. It's been fantastic. So thank you. As a reminder to everyone. Oh, Ben, do you have something? Yep,
Just
go for
quick
it.
mention, looks like there's many PhD students and then who are interested in this career path. The LGG fellowship application cycle just started, even though our Vanderbilt does not accept any applicant for this year, but you can find the application information their fellowship website. And then also, Kaylin already posted in here, LGG info is kind of looks like cool website to provide a kind of answering all of your question already, how to prepare this fellowship and how to prepare a fellowship application, how to stand out from other candidate type of things. So please check out.
Yes, excellent resources, and thank you, great manager. I've been trying to put some of them in the chat as we go along, so they're available. I put in the link if you want to learn more about volunteering for ClinGen, which I'm glad there are so many people interested in that. I just put in the link for the karyotyper that Dr. Smith mentioned, and as Dr. King mentioned, I put in a few of the links to the websites that he provided in terms of individuals wanting to become a strong applicant for the different fellowships moving forward. And the last thing that I encourage everyone to do as someone hosting these webinars is there's a lot of organizations that do host webinars for you to learn more about these different fields, to hear from experts in the field. Again, it could be a networking opportunity, or it could just spark an interest in terms of, that's something that I really want to do, and perhaps connect with the speaker or connect with the program that's doing that. And that shows a lot of interest and can help pave some pathways into getting experience and getting some knowledge as you learn more about what you may want to do. And I think all of the panelists here have kind of carved their own way. So, just because there's a fellowship pathway doesn't mean that's the only way for you. And there's definitely different jobs and different side steps and different things to pursue the passion, and it will come out in different ways. And as Alexa had mentioned, she carved her own career, carved her own job. So don't just because there's a fellowship feel like that's the only path or the only box as you move forward. So thank you again to our panelists. I am going to just share my screen one more time just to indicate that we have a few more of these sessions coming up this summer. Again, this is the ClinGen Clinical Genomics Career Webinar Series. This is the 4th year. The recordings from the other 2 years are also on the other 3 years, sorry, that's math. The other 3 years are also on our YouTube playlist. You can find the link on the ClinGen website. Here, this green box is where you get information on the Clinical Genomics series, and this box on the side with the 2 people on the desk around the world is volunteer to curate. That's where I've been sending you all if you're interested in learning more about curating. And so thank you all, and we will be sending out a feedback survey at the end of the last session, and so just gives us a little bit guidance on how to direct these sessions for the upcoming year. So again, a massive thank you. I really appreciate the panelists' time, really appreciate your expertise, and I hope everyone has a great day, and if anyone's interested, we'll see you next week.
Thank you.
Thank you.